Hey guys, my name is Hannah Patel, and then today we'll be talking about DNA replication, translation, and transcription. First, let's talk a little bit about DNA. What exactly is DNA? DNA is a deoxyribonucleic acid that consists of nucleotides. The nucleotides have bases on them. You have 4 bases: adenine, guanine, thymine, and cytosine. Your adenine base pairs up with your thymine base, and your cytosine base pairs up with your guanine base. But keep in mind that in RNA, there is no thymine, therefore thymine is replaced by uracil, which results in adenine pairing with uracil and guanine pairing with cytosine. Alright, with that, let's get started on replication. So, replication. Replication is when the double helix is unwound, giving you 2 strands, your leading strand and your lagging strand. And then whenever it's unwound, you get a little replication fork. And then your 2 strands, your leading and lagging, they will act as templates to make new strands of DNA. On the leading strand, you have a primer that will bind to it. The DNA polymerase is the primer that binds to it, and it will bind along the leading strand and add new complementary bases to it, and therefore that makes it continuous, as you can see here. Your lagging strand is not so lucky, and it does not get that. So your lagging strand has Okazaki fragments, which are these little breaks in between the strands, which makes that discontinuous. And therefore, whenever everything is paired up, the bases are paired up, your primers will go away. Then it will start to proofread itself to make sure everything's right where it's at, everything matches up, there's nothing out of place. After it's proofread, your DNA ligase will come in and basically glue together the sequence of the DNA. And that is replication. And then after that, we will move to transcription. Transcription consists of 3 steps: your initiation, elongation, and termination. In your initiation, RNA polymerase binds to DNA. The RNA polymerase attaches to a specific area called the promoter region. The promoter region is generally upstream. And then we move on to elongation, where the transcription factors unwind DNA strands and allow RNA polymerase to transcribe a single strand of DNA into a single strand RNA polymer called mRNA. After the RNA polymerase moves along the DNA, it will keep going until it reaches a termination sequence. Once it hits that termination sequence, your RNA polymerase will leave and it will detach from the DNA, and transcription stops. The very last thing is translation. So translation occurs in the cytoplasm. The biggest role player in translation is transfer RNA. So your mRNA binds to a specific site on the ribosome, and the ribosome starts matching tRNA codes on sequences to the mRNA code sequences. Each time a new tRNA comes into ribosomes, amino acids get added along the polypeptide chain. And then this continues until you hit a stop sequence. Once you hit a stop sequence, your polypeptide is released and the mRNA is released, resulting in tRNA to bind to the ribosome, and that is where translation stops.